Good day. My name is Carlos Mota, and I'm an assistant professor at Maastricht University in the Netherlands in the south. Today I'm going to speak about 3D printing and bioprinting of in vitro models and implants. So we are located in the south of the Netherlands in a very small city called Maastricht, a riverside city, very nice to visit, so you are welcome to come and visit us. We are located in the Faculty of Health, Medicine and Life Sciences of Maastricht University, and this is part of the healthcare campus. So we are embedded within the hospital. We are a young institute. We started 10 years ago in 2014 from an institute that was in the north in the University of Twente in Enschede, but we have established a new group at the University of Maastricht from 2014. So today I'm going to speak about 3D printing activities that I have in my group, but also bioprinting activities. And I will try to explain you a bit the differences between the two and why we use these technologies. So we start with a bit of a general introduction of 3D printing. So 3D printing has been there for almost 4 years. So this is a patent from Charles Hull, which patented a technology that was able to produce three-dimensional objects with a resin. So as you can see here in the diagram, you can use a laser to photocrosslink a resin in specific areas, and then if you repeat this layer by layer, then you can create an object. So this was back in 1986, so a lot of new developments have now arisen around stereolithography, but this is after the patent was expired. This is one of the technologies that we actually use in the lab, but it also took a lot of time to be integrated within tissue engineering and regenerative medicine. So these technologies have been embedded or applied to healthcare, but not directly. So early on, the 3D printing technologies were applied indirectly. So here is an example where 3D printing was used to create phantom, so objects that emulate tissue, and in this particular case, they were used to then train surgeons. So in this case, you can see that this tissue can be sutured, so this can be quite handy to then treat surgeons or doctors that are under training. Other applications was, for instance, to create anatomical models, and this was to also simulate complex surgeries. So in this case, was a 3D printed section of two fused Gemini that were separated, and then the idea here was to have a bit of a dimensional object that then the surgeons could plan ahead the complex surgery to try to reduce time, but also to try to prevent complex cases or complex situations during the surgery. More recently, there is more direct application of 3D printed objects in clinics. So in this case is an example of a custom-made jaw implant. And normally these 3D printed objects that are in the clinics are normally made of stainless steel or other alloys, because they are, inert and they will not react with the tissue, and it's much easier to bring this type of implants to clinics. So this is actually a spin-off company from our university and our faculty that now creates patient-specific implants with selective laser melting, for instance, or also with subtractive technologies for clinical cases where the typical off-the-shelf implant doesn't do the job. So with regards to 3D printing or additive manufacturing, so normally we call it additive manufacturing. 3D printing is more frequently used in other fields and also among the public, but we normally call it additive manufacturing. We use it quite a lot for tissue engineering. So what is tissue engineering? So the principle of tissue engineering is to offer a temporary scaffold or a temporary structure where this temporary structure can facilitate the regeneration of tissue and promote the new tissue formation. So if, for instance, we start here with a medical imaging, So we can acquire data from the patient directly from MRI or CT scan. Then we can convert these images into a CAD model where we can identify the defect. We can, of course, discuss with the surgeon if that defect needs to be adjusted or if there is tissue that has to be resected, and then plan an implant or a temporary scaffold that can be custom fitted to the patient. So in this case, we then use additive manufacturing to produce scaffolds, and these scaffolds normally are highly porous to facilitate the new ingrowth of new tissue, either bone or soft tissue. And afterwards we have a step where we normally seed stem cells. So we use normally bone marrow stem cells, but also other stem cells that are seated on top of these scaffolds, and they are then cultured in vitro to then evaluate if the scaffolds promote or allow the formation of new tissue. They can be either cultured statically or dynamically. And then after the implantation, of course, the expectation is that you have a fully regenerated defect or area that has been initially affected. So we use several techniques. Here you have, for instance, stereolithography, which was the first patent, as I mentioned. The drawback of this technique is, was in the beginning that there were no non-cytotoxic resins. So normally the resins that were used industrially were toxic for cells. But gradually chemists have been developing new resins that are more cytocompatible, and it's nowadays possible to use stereolithography to produce scaffolds with cells embedded. So and that is normally called bioprinting. I will come to that in a couple of slides. We also use selective laser sintering. So the example that I mentioned of the company that came from the faculty. So they use selective laser sintering or electron beam melting to produce metallic scaffolds. So in this case, you have a metal powder that is selectively sintered with or without an adhesive with a laser. Three-dimensional printing. Normally, three-dimensional printing initially was identified as the same printing technology that you would have in your office, but then the difference is that you would have a powder bed that would gradually allow to create a three-dimensional object. And this powder bed was selectively binded with a binder agent, which normally was toxic, and that binding agent would be pyrolyzed, so it would be burned away from the object, and then the object would shrink. So normally these objects are quite rough in terms of surface porosity. It depends on the particle size. So it's not a technology that has been used quite a lot on tissue engineering. We use quite a lot fused deposition modeling, and this has a typical 3D printer that you use at home extrudes a polymer melt, so either a polymer or a combination of polymer and in an organic filler. The only difference that we have in our labs is that we don't use filament coils, so we normally use medical grade polymers, and normally those come in pellets or powder. But I will come to that in a couple of slides. We also use solution or slurry extrusion-based techniques. So in this case, you have, for instance, a polymer which is partially dissolved in an organic solvent. This is, for instance, to create highly porous structures. Tissue and organ printing, I will come to that. Additive manufacturing, wet spinning. Again, you start with a polymeric solution, for instance, and then you phase invert that solution into a non-solvent solution, so that then you also have a porosity inside of the scaffold. We also use quite a lot electrospinning, melt electrospinning, or direct writing electrospinning. And this allows us to produce nano-sized fibers to emulate the extracellular matrix of the tissue. So, for instance, this is selective laser sintering. Here you see the laser selectively bind particles of powder, but as you can appreciate, normally these objects are quite rough, so depending on, of course, the particle size. But one of the technologies that we use quite a lot, as I mentioned, is fused deposition modeling. we can then create scaffolds. So you here on the bottom right, you can see a scaffold that is highly porous, and then what you see a bit of more brownish is cells that were seated on top of the scaffolds, creating a new tissue. So we started looking at how we can emulate tissue and how can we create scaffolds to emulate tissue and to emulate also tissue regeneration that naturally occurs. So when you have a bone fracture, if the fracture is small, normally the bone will regenerate by itself if the fracture is kept in a close proximity. And what normally happens is a continuous cascade of gradients that allow this tissue to be then formed again. So you can see there is a callus being formed, there is vascularization occurring, and then normally gradually this bone will heal and the soft tissue, the callus tissue that is formed, is then reconverted into bone. So of course that, in the magical world, if the fracture is in the close proximity, then it heals. But then if you have a long bone defect, if you have, for instance, a tumor that was resected and there is a lot of space to fill in, normally the bone doesn't regenerate. And to circumvent that, surgeons use autografts or allografts or demineralized bone matrix, but all of these have quite a lot of constraints. For instance, autografts, you have donor side morbidity, you have limited availability, but you also have other drawbacks, let's say. So we want to use fused deposition modeling to create scaffolds. And the advantage of these scaffolds, as I mentioned, is that you can design the shape matching the defect. You can make these scaffolds fully interconnected. You can control the porosity, the pore shape, and the pore size. So if we look at bone has continuous gradients, not only in terms of porosity, but also mineral content, for instance. So we wanted to try to emulate this. So we started early on to try to emulate this by looking, for instance, at creating gradients of porosity. So here you have a radial gradient, emulating a bit more the trabecular and the compact bone on the extremities, but also other gradients. For instance, when we combine different materials, then you can have different stiffnesses. So for instance, POT PBT is a bit softer and then PLA is harder. So you can create, for instance, stiffness gradients and wettability gradients as well, because they will behave differently in terms of protein adsorption to the surface. So, but as you can appreciate here in these images, these are discrete gradients. So they are staircase, they are not continuous. And this brings drawbacks of delamination, for instance. So we took a bit of inspiration of Franz Carlier's statue here in Maastricht. So if you look at the human body closely, it's not a staircase. It's not a discrete gradient. It's a continuous gradient, actually, but at different scales. So what we wanted to circumvent was the discrete gradients that we have been achieving up to that date, and then we wanted to do continuous gradients. So we wanted to also take a bit of inspiration on nature, to different tissues have different gradients, but then when you have the bone fracture, there is a lot of gradients occurring. So we had this technology, we wanted to have this technology because we wanted to play with structure, but also composition, and also the combination of two. And as I mentioned, we wanted to do a continuous gradient because in our vision, it would be better to bond at the interlayer interface, but it would also mimic the in vivo microenvironment. So we designed a new fused deposition modeling technology, which allow us to dispense then two different materials, and then these two different materials could be compounded with each other, so we could mix them. But at the same time, we could also activate the surface, because one of the drawbacks of thermoplastics is that normally the cells will not immediately attach to them. So we designed this print head to allow then mixture of the two materials that were in the two reservoirs. We had to simulate a bit the mixture and try to design a screw that would allow us to do that mixture. But you have to understand that doing a small print head that will fit inside of a commercial platform, a commercial printer, is quite challenging. So to give you an idea, this mixture between the two materials happens in a screw that has 6 millimeters of diameter and 40 millimeters of length. So there is a bit of limitations in terms of mixture, but we are able to mix between the two compounds. We also did some computational modeling to try to predict a bit the level of the mixing. And since the technology works with polymer melt, another important aspect is to have then control on the temperature of the different reservoirs, but also on the mixture, on the mixing chamber. So we did empirical testing. We did empirical testing for a lot of different materials. For instance, materials with compounded with reduced graphene oxide, with lamellar ceramic fillers that were used to protect antibiotics. So we also had scaffolds with antibiotics, for instance. So, and by playing a little bit with the pressure differential between the printing reservoirs, then we could produce these scaffolds. So I will show you now the video of the system operating. So if we then apply different pressures to the different reservoirs, we are able to dispense a material A or a material B, and then applying different pressure differentials, then we can then extrude different ratios and have different mixtures happening on the screw of the extruder. So as you can see here on the color, you have a continuous gradient radially, but also here when you have a black and white contrast, and this is then controlled by applying different pressures and different pressure profiles. So again, we wanted to look at bone fracture and create gradients to heal bone fractures, long bone fractures. So what we thought of was to create osteoconductive scaffolds and osteoinductive scaffolds. So osteoconductive scaffolds, you would have a higher concentration of ceramic, in this case was hydroxyapatite, and then a lower concentration of ceramic in the center. So as you can see here, and then also an osteoinductive, where we had more inorganic filler, hydroxyapatite, which is bones are composed of a huge percentage of inorganic phase, and we wanted to have this inorganic phase in the center to allow the cells to then be recruited towards this central area. We also had the controls, and then we implant these scaffolds in the ulnar defect for 3 months. And then when we did the explant, we saw that there was a good integration. So here we cut the radius, and then we pull apart and then push the scaffold. So you see that there is a good integration of the scaffold with the surrounding tissue. And then we also did some micro CT. There is new tissue formation, so and it's also depending on the type of gradient that we use. If we use a negative control where we don't have a filling or a scaffold, normally what happens is that extremities of the ulna will fuse to the radius, and then when you have a positive control, which was collagen sponge with BMP, then you have a aberrant bone being formed. When you use the scaffold, then there is gradually new bone being formed, but of course this is still a bit suboptimal because the explants were made at 3 months, so we don't have an abundant amount of bone within the scaffold, but we have some bone being already secreted inside of these constructs. So this was also confirmed by histology. We are trying to quantify this throughout the length of the scaffold, but we also see that there is the natural effect of having a bit of more callus formation, so soft tissue being formed inside of the scaffold first, and then this soft tissue being then mineralized over time, so that then you have a new bone being formed over time. So we also wanted to use atmospheric plasma, and we want to use that because if we use thermoplastic polymers, normally the scaffolds, the cells will not immediately attach to the surface of the scaffolds, and this is quite important for new bone formation. So as you can see here on the left, if you don't treat the scaffold, then all the cells that have been strained in green gradually settle because of gravity. But then when you treat the scaffold in the right side is with atmospheric plasma, so you treat the full scaffold all at once, then the cells attach to the surface quite well. So we wanted to incorporate this into the printer because we also wanted to create gradients. So we had also quite a lot of simulations to optimize the atmospheric plasma. So what we could achieve was the same type of gradients. So gradients where we had two extremities that were activated more with plasma, so that where the cells could attach better, and then the central area without activation, or the extremities without activation and then the central area with activation, or the full scaffold being treated. So here you can see the manufacturing in operation, both the continuous gradient. So you see here a scaffold that is being produced with a gradient of polymer and polymer that has been dyed with a red dye for simplification. And then you see the atmospheric activation in the middle to activate a very selective portion of the scaffold. So this would be, for instance, important for us to have different populations of cells. So one of the limitations of these very big scaffolds is vascularization. So here we have a scaffold that was activated in the central area. In this case, we could see a cell population that was stained in green. For instance, we could see the UVEX, and then have the UVEX attaching selectively in the center of the scaffold, and then having, for instance, bone marrow stem cells attaching all around the scaffold to have different localization of different cells within a construct. So with this technology, we were able to do different structures, composition, play with a bit of structure and composition, and also functionalize the surface. And all of these conditions can support better bone formation. But of course, this is still work in progress. You might think that some of these technologies are far away from clinics, but reality is that there is already some clinical examples. These are not scaffolds produced by us, but are scaffolds produced by a company in Singapore that uses the same technology, uses PCl with also tricalcium phosphate. And this is one of our clinicians from MUMC, from the Maastricht University Medical Center, then applying some of these scaffolds for long bone defects into patients. So as you can see here, the surgeon is coating the scaffolds with bone marrow extracts from the patient itself during surgery to try to facilitate or to improve the regeneration. So with regards to bioprinting, 3D printing, we normally start with a scaffold and then we see the cells on top. Bioprinting is different. So bioprinting introduces the cells already during the process. So when you produce a scaffold or when you produce a cellularized construct, the cells are already in the system. So there's a lot of definitions out there. In 2018, there was an effort from us and from the International Society of Biofabrication and some members of the society to try to come up with a definition for bioprinting. You still see that is a computer-aided transfer process, and you see that is for patterning or assembling of living and non-living materials. So, but in my perspective, normally bioprinting always introduces the cells within the process. So there is, again, similarly diverse types of bioprinting technologies. We have pressure-assisted bioprinting, which is normally the typical bioprinting technique because it's easy. You can use disposables, so you don't have to be concerned about sterilization because you can use disposables that are sterile and cross-contamination. And it's quite cheap. So nowadays these extrusion-based or pressure-based bioprinting technologies are quite accessible to many researchers worldwide. Also, you have inject and droplet on demand. In this case, it's the same technology that you have in your office printer with some peculiarities. So in this case, there are several different techniques to produce a droplet. So in this case, is a droplet by droplet. In the previous case was a continuous extrusion layer by layer, but in this case is a droplet by droplet, and then you have different actuation modes: thermal, piezoelectric, electromechanic, and also acoustic waves. So you can create a droplet and then displace this droplet between the print head and then a substrate. the throughput of such injector droplet on demand is low, so it takes some time for you to produce a three-dimensional object. As I mentioned, there is also stereolithography, which has evolved quite a lot lately, because there has been new developments in terms of biocompatible and biodegradable resins. Laser-induced forward transfer uses a laser to create a droplet and to transfer cells from a donor slide to a target slide. Again, a very high precision technique, but with a low throughput, or a relatively low throughput. And then you have microfluidic bioprinting, where you can pick a bit advantages of the microfluidic field, and then use this very well-known field to pick a bit of an inspiration of print heads that allow the combination of different fluids. So then you can create a filament with different configurations of materials and materials with cells. So the advantage of microfluidic bioprinting is that you can have, for instance, a shield fluid, which will prevent a bit the shear stress, which is normally one of the drawbacks of most of the bioprinting technology. So this shear stress will induce, for instance, cell death. So we have been working quite a lot on kidney, but also other applications. Of course, that we would envision in the future the possibility to create tissues for regenerative medicine purposes or for implants. But nowadays we are mainly focused on in vitro models, and this in vitro models will allow us to evolve on the technology side, on the material side, but also on the biology side, and then to try to see what we will need in the future to create a fully functional tissue, which could be then implanted in the patient. But we are still far from achieving that, especially on the case of the kidney, which is quite complex. So for the bioprinting, we normally use a bioink, and a bioink normally is a combination of hydrogels and cells, but can be also a combination of hydrogels and growth factors or other small molecules. Normally a biomaterial ink is more associated with additive manufacturing, so when you have a scaffold that then is seeded with cells on top. So this falls a bit under the additive manufacturing criteria that I showed you before. But normally with bioprinting, you normally term a bit what you use to process in these bioprinters a bioink. So what is an ideal bioink? A bioink normally has to suit a lot of purposes. It has to be compatible with the bioprinting process, but also allow the cells then to take over after the bioprinting process. So normally the bioink needs to be designed to protect the cells from shear stress. So normally you use materials that are shear thinning. To allow mass transport or diffusion, should interact with the cells in most cases, and then you have also bioinks that are designed with specific mechanobiological cues, biochemical cues, but also these bioinks need to be designed with the possibility that the cells then degrade these bioinks, these materials, over time at the rate that the cells dictate. So there's also new bioinks being developed in the field. There's dynamic bioinks, and these dynamic bioinks allow, for instance, to respond when the cell pushes a material to dynamically rearrange over time. So this is a new type of bioinks that are being investigated to try to facilitate a little bit more the cells to then take over after the bioprinting process. So, as I mentioned, we use these bioprinting technologies to study mainly in vitro models, but of course with a big vision in the future to create implants or regenerative medicine strategies. But I will show you a bit on where we started with the in vitro models. So as I mentioned, we work quite a lot on kidney. So kidney is a very complex organ. It's quite amazing organ. It continuously filters your blood non-stop. And the organ is comprised of millions of nephron units. And these nephron units are composed of more than 20 different cell types. And the different cell types that are composed in this nephron have different functions. So, for instance, as you can see here, you have the Bowman's capsule, And as you can see already in this very small compartment, there is multiple different types of cells that allow, for instance, to do filtration, the primary filtration within the Bowman's capsule. So we had quite a lot of challenges to think of, which cells are we going to start with, which biomaterials, but also which bioprinting technology we would start. So as I mentioned, we had the possibility to acquire a microfluidic bioprinter. And the advantage of this printer is that it allows us to reduce the shear stress when printing, because one of the materials that we use on the outermost part of the nozzle is normally a shield fluid that prevents the cells to be then sheared against the surface of the printing head. So here you can see the printer working. So we normally print with with a transwell membrane, so that then we allow to remove that shielding fluid. So as you can see here on the right side, we have a filament being formed, and then we have the shield fluid dripping on the surrounding. So this shield fluid acts like a lubricant, more or less. And then you have an accumulation of the fibers, because in this case it's not directly printing, it's just producing the fiber itself. So we started with primary cells from mice. Mice are quite useful, because you can also generate mice which carry reporter lines, but you can also have disease model mice. So we can isolate these cells. So we isolated fibroblasts, tubular epithelial cells, and endothelial cells. We have optimized not differentiation protocols, but expansion protocols to culture these cells in sufficient numbers for us to bioprint. And it was possible to culture mainly the epithelial cells. The endothelial cells was a bit hard to culture them and to expand them, and the fibroblasts were relatively easy to culture, but normally the fibroblasts would overgrow quite fast. So we also characterized these cells. We also had an in vivo mice model, which was healthy and fibrotic. So fibrosis is one of the pathological endpoints of patients with chronic kidney disease. So the idea was to use the two different cell populations to then have an in vitro model which could emulate the healthy condition, and another in vitro model which could emulate the pathological condition. So we picked these cells, and as I mentioned, the epithelial cells were easy to expand, and we combined them with endothelial cells. So we use human vascular endothelial cells. So we had the two different cell populations being printed into a core shell construct inside of a filament. So after 14 days of culture, you already see a bit of rearrangement of the cells inside of the filament, and then at a certain point you have a kind of a lumen being formed inside of these filaments to try to emulate a little bit the proximal tubule. So this is just a small little portion of the nephron unit, so the functional unit of the kidney. Of course, this is a very simplified version, but it will allow us then to study, the healthy condition and the pathological condition, how the kidney disease progresses over time. So we are still working on this in vitro model, and we are having some degree of success on emulating the pathological but also the healthy condition. The caveat here is that you are just emulating a small section of the nephron. So if we want to look at the complexity again of the kidney, normally the kidney is formed during the developmental stage out of two different progenitors. So normally the kidney is formed from a metanephric mesenchyme. So most of the kidney, the cortex area, is formed by this more red population. And then with the crosstalk of ureteric blood population, then there is the gradually growth of the kidney. So this can be emulated with kidney organoids differentiated from induced pluripotent stem cells, and we have established these protocols in the lab to then create these two progenitors, so that then we could emulate a little bit of the development stage of the kidney. During this differentiation, it is possible then to obtain more different types of cells that are comprised in the nephron filtration unit. So here you see one of the populations, the metanephric mesenchyme. When you culture them over time, then they start to form structures. So these are structures that are emulating a little bit what happens during development, but it's not controlled. So you cannot really dictate where the cells will rearrange. So here you see some structures stained in green, which is the proximal tubal portion of the nephron, but also podocalexin, which is the red staining, which emulates a bit the Bowman's capsule, the podocytes cells within the Bowman's capsule. But you also see CD31 in yellow, which is a bit of a rudiment of a vasculature. The other progenitor is ureteric bud. You can see here a confocal where you have the two different markers, cytokeratin 8 and GATA3, which are two of cell-specific markers for more distal tubal collecting duct part of the nephron. So we wanted to pick these two populations and then combine them together to try to improve a little bit on how we can achieve these organoids. So we have also used the microfluidic bioprinter to print progenitors. So you see here the single cells printed inside of a fiber, which will gradually reorganize and create these clusters of cells. And then after 7 days, you can already start to see the structures. And then after 12 days, you can already start to see different organoids. And then when we stain these organoids, you can see a good arrangement of cells, but you can also see the specific markers for proximal and distal portion of the nephrons. So this is quite interesting. We are still working on this project. We wanted to then be able to combine the different progenitors, but then also have different ratios of progenitors to try to emulate a bit closer what happens during development. We also have an European project following up on the combination of bioprinting and these iPSC-derived organoids and iPSC-derived cells with organ-on-chip. So one of the limitations now of these organoids is that once you culture them in vitro, then they will gradually degenerate because there's a lack of vascularization, there's a lack of maturation. So we want to culture this into chip-on-chip technology to allow them to mature them a little bit more, but also to allow to use them as an in vitro platform to then test drugs for a longer period of time. In this specific project, we want to emulate BK viral infection and also nephrotoxicity. So we are using the combination of these technologies to then have more physiologically relevant models. So we use a very specific bioprinting technology. Again, is a microfluidic bioprinting, but then in this case, it allows us to pattern really with a very high accuracy single cells. And then we are redesigning a commercially available system with tissues, a company that is part of the consortium, to incorporate then the bioprinted construct within the chip model. So with regards of bioprinting, of course, people are looking into different organs and tissues. We also are looking, for instance, into cardiac tissue, into thyroid, into osteochondral implants for cartilage and bone regeneration. So we have different tissues and organs that my research group and also the department is looking at. But if you want to read more about it, this is a review that we have written back in 2020, which has a very extensive description of what are the models that are developed, what are the tissues and organs that researchers are targeting. If you think, okay, bioprinting might be a bit far away from clinics, just because it's not, a simple scaffold, a simple or a simpler medical device. It's more classified as an advanced therapy because of the inclusion of cells. It might be far away from clinics, but reality is that some of the examples are already being tested in clinics. So one of the first examples that will most likely reach the clinic soon is skin. So what you see here on the right side is a comparison between the bioprinted skin and the human skin, the human native skin. So there is some resemblances on what can be achieved with bioprinting in terms of tissue. So this is most likely the first application of bioprinted tissue in the patients in the future. There are other applications, for instance, from the central nervous system, peripheral nervous system, bone tissue, tissue itself, for instance, heart tissue, again, a very complex organ to emulate with bioprinting, but the researchers are focusing also into that. Liver, pancreas as well. So we have also some work done internally in pancreas, mainly to print islets of Langerhans, which is one of the functional units to then metabolize insulin. So this is some of the work that we are looking at. There's also researchers that are highly focused on vascularization, which is one of the main challenges of bioprinting, because if you use a very highly cellularized construct with cells, then they will immediately need to be fed, so they need to have access to nutrients really fast. So then researchers are also looking into vascularization. So with this, I would like to acknowledge all the contributors of the work, all the team members, all the collaborators from the different projects, and the funding agencies, the Dutch Kidney Foundation, for instance, that funds all our work on kidney, but also all the other European grants that fund the diverse projects that we have ongoing in the lab. So with this, I would like to conclude. Thank you all for your attention. If you have questions, do not hesitate to ask or to send me an email by reaching out to the email that you see on the bottom left. Thank you very much.